Yeah. Basically, we're gonna try to be a bit broad here and cover some aspects of yeah like the overall ethical concern behind the stem cells, but also like the clinical application. Some example on which types of disease has been experimented on, and yeah, like how far we got. But that paper, I wrote it with okay, so I cannot see those the slides. Can everybody see the slides? Or like the
That's
question
amazing.
is, okay, I'm gonna jump out and jump in quickly. Come on, come on, Discord. There you go. Okay, I see it. It's perfect. Okay, so this paper was written in December 8, 2012 by Lasse Baltersmith, Zinja Antar, and myself, with supervisor Jette Rank in a third semester project. We worked together to bring in an understanding of the importance of stem cell. And it was December 2012, so most tinfoil hat were believing that the world was going to end and everything. So it was probably not the best time to hope for such a thing. But luckily, it doesn't discourage some good scientists for continuing to build up and debating empirical evidence. So Yamanaka kept up with well, I don't yeah, I don't think he went as far as realizing his vision of a induced pluripotent embryonic stem cell bank, but he got close. I saw some recent paper on his cocktail still being used and showing promising results. So the Roskilde University slogan, I don't know if you see it, but it's also a good introduction to how important the Latin is. In calmness and silence, there is death. In the flow of a wave, there is life. Basically, it's not useful to just sit down and think, okay, that this is illegal or the ethical committee doesn't approve it. There's always a way of debating and um, breaking some taboos or overcoming some conservative views and so on. So I still live in Roskilde, and even though I have yet to write my bachelor, my motivation for this specific field is still as an all-time high. I'm thinking above simply being a college dropout, and I'm gonna do what I can to promote and contribute to scientific advances regardless. I will keep up. So let's get to it and analyze this paper. So we have an abstract that says the experimentation and use of human embryonic stem cell and inducing pluripotency into adult somatic cell is a topic that is highly controversial and discussed. We strive to convey and clarify some of the concerns. The paper we wrote is an attempt to get the definition and characteristics of those stem cell as well as their respective harvesting methods. Two example of application in clinical therapy is presented, showing that stem cells could possibly cure Parkinson's disease and diabetes type 1. Human, like adult stem cells, has been through many ethical issues and that have prevented further discovery and clinical application in the clinical application in the treatment of human disorders, which potentially could have saved many lives. But many misconceptions, conservative concerns, religious beliefs, and corporate interest are to be surmounted or circumvented in order to improve future treatment and therapies. Yeah, the next slide. So the introduction covers that embryonic stem cell derives from differentiated totipotent stem cells. They divide and differentiate to specialize and become either of the three germ layers: ectoderm, which is the outer layer; mesoderm, the middle layer; and endoderm, the inner layer. So look up Wikipedia for further explanation and details. The TLDR is that these layers give rise to all tissue and organs of most animals through organogenesis, and the potential of these fetal and embryonic stem cell make them highly valuable. Deriving adult stem cells into induced embryonic stem cells to cure diseases and regenerate tissue, as well as recreating entire organs with maximum gene compatibility, could theoretically be accomplished given the right conditions. Therefore, curing a myriad of diseases such as Parkinson's or diabetes by using stem cell therapy as an example, we cover in our report without well, it is assumed to be possible without telomeric or genetic issue that may happen using other types of cells. Adult stem cells are known to yield chromosomal and telomeric integrity, and it could therefore theoretically be possible to repair and restore function in any given microenvironment. Yet we also want to avoid the ethical and legal limitations, and that's sort of where Yamanaka's vision of an official induced pluripotent stem cell bank is. Okay, it's basically like you have your specific adult stem cells that are of specific types, and Yamanaka back in 2012 he wanted like a huge repository of the different stem cell type in some sort of a bank of like a stem cell bank, and it is still unacknowledged or directly interdicted in some countries to have induced pluripotency or like playing with it because genetic engineering and so on. So yeah, the next slide.
Hang on, quick question.
Yeah,
can I get a show of hands here? Who knows what the word pluripotent and totipotent mean?
Me.
Pistachio?
Yeah, I'm familiar.
Nope.
I think I do.
Cool.
Cool.
Can Yamanaka, can you give us like a really quick breakdown of that so that everyone has a little more background?
Of course. So adult, well, totipotent stem cell comes from an adult stem cell derived from the bone marrow, or they're present everywhere, but usually, like typically, they're mostly present in the bone marrow. So totipotent stem cells are those that can differentiate into their like into the specific site they're at. Like if it's from the bone marrow, they'll cover those the cells around, and as we'll talk about it later, but there is like a threshold on normal cells, like on non stem cell types. They can only differentiate uh 40 to 60 times, and then they break down and are told to basically go apoptosis, well, like basically terminate themselves. And that's where we have like adult stem cells, which is totipotent stem cells that constantly produces daughter cells that are like tissue specific. Pluripotent stem cells, on the other hand, comes from the embryonic. They're they they've only been differentiated from 4 to 8 times, and they can become a much larger. Well, they can specialize in a very like in everything we're composed of. Like they can be in anything theoretically. Uh, so therefore, we have like this concept of inducing pluripotency into adult stem cells. The acronym is IPS, small I and PS, or IPSC, and those have like gone back in time, sort of say, to adopt pluripotency again. Like they're basically told to become embryos, like you would. I forgot the name of the hormone that does it in plant. Is ibonucleic? It's like it's basically like the plant hormone that makes them germinate again. Those factors, the Yamanaka factors, they can basically tell an adult stem cell to become embryonic. Does it cover the question?
I would say so, yes.
Cool.
Yeah.
Thank you.
So okay, yeah, the formulation of a problem. This is probably the most important part to figure out when you're willing to write a scientific paper. The main questions that your research attempt to discover, clarify, narrow, detail, prove, or contradict are formulated in such a problem. Like it's a good set of questions that should be answered, or at least should be uh, demonstrated. Like the way you would go about answering those questions should be demonstrated in the report. So our problem formulation was what are the main characteristics of different stem cell type? What are their clinical and use and ethical concerns? Which application could human embryonic and induced pluripotent stem cell be used for in our current medicinal industry? And how could the pros and cons of using human embryonic stem cells and IPS be debated? Those are the questions that we attempted to answer, and let's get to it. Um, Next slide. Characteristics of human embryonic stem cells. So pre-adult stem cells are characterized by their high telomerase activity and the ability to produce all types of cells from the 3-germ layer. High telomerase activity of pre-adult stem cells remain unaltered, although through differentiation stages, telomerase activity is lost and adult cells eventually reach a differentiation threshold called the Hayflick limit. So the Hayflick limit is basically saying that it can only replicate and divide 40 to 60 times before it cannot divide anymore and will break down by programmed cell death. Under healthy circumstances, stem cells keep dividing until loss of telomerase or if the DNA is not intact anymore. So precautionary measures such as bisulfite sequencing and other genome sequencing techniques are to be performed to ensure ideal conditions. Pre-adult stem cells self-renew and differentiate to the multipotent stem cell that could be used in regenerative medicine as long as the human leukocyte antigen HLA matches the patient own genetic profile and the embryonic stem cell is derived in a proper way. So the figure one that we're about to see shows the development stages of a fertilized egg and how it's converting into pluripotent embryonic and then modified into the required organ part of the patient. Bear in mind it's kind of an old graph. So yeah. Anyways, next slide. So as you can see, from the fertilized egg, marula, blastocyst, pluripotent, and embryonic, you can just make them specialize into any tissue organ given the part of the patient it would be. Let's say the bone marrow, because high telomerase, and you apply the nana cocktail in specific way that it becomes pluripotent but also still like very tissue specific. So you basically you cut the route of the fertilized egg, marula, and blastocyst, and instead you take out a totipotent and you directly introduce it. Well, you induce pluripotency and you directly introduce it to which tissue it should specialize. Um, yeah. Any questions so far? Well, we'll take them as they come. So yeah, from the book that I enjoy using, you have the one to the right that are like mostly adult stem cell that produces blood. So yeah, or antibody lymphocyte, and yeah, and to the left you have those that, like, you have the embryonic that can differentiates into many types of blood cell, but they are also the only one capable of becoming nerve cell or liver cell or like organ cells. Adult stem cells, you can like, without inducing pluripotency, you can tell them to become blood cells, but not really to become nerve cells or other type. uh any questions on that?
I'm actually a little curious here. Have you seen
Yeah.
people actually do this, like where they actually make blood cells, or yeah, I guess blood cells first.
Well, I live in Denmark, so therefore Europe. So I haven't really seen it with my own eyes. We've kept it like theoretical all the way, and yeah, I have yet to travel the world and see how it's done in practice. But for now, like, we're restricted. Like, the ethical committee of Europe,
I
they don't. Yeah.
that's it. That's all I've got. Yeah, I've seen it.
Okay. Yeah.
Yeah,
You've seen
it's just
papers
like
on it,
it's
or
no, it's just like
Nice.
research work that I did.
okay.
Yeah,
Cool.
it's not really like paperwork in as much as that it was just like replicating other work. We were working on a completely different project that had to do with 3D printing capillary systems and then implanting stem cells into them. So it was completely a side thing.
Mm.
It
Okay.
works. It's a thing. Yeah.
Okay.
I guess I'm
Not-
asking just because I've seen it. Well, there's a company local to me. I'm in Canada. I'm near Toronto. There's a company local to me that does. They have like a company, and allegedly they're making blood cells and doing fake or synthetic blood, but they've only ever done it like at really small scales. Like they've never actually made like it in significant quantities. And I think I've seen a couple of companies do it that way. And yeah, I was kind of curious to know if anyone had seen it work like in a more practical
the
No,
way.
good
scaling
question.
the
Yeah.
scaling is very difficult for this because
Yeah.
I as I've mentioned before, if anyone's heard any of the the biohack chats, stem cells differentiate due to mechanical, chemical, and physical forces, and so that's like movement, structure, and chemicals.
The inductions.
Yeah, exactly. And those are like your three primary inductions. And so when you scale, you actually, it's like when you're cooking. You know how when you make a small dish, it cooks in a very specific way, and then when you make a big dish, like all of a sudden you're like everything is soggy. What's going on?
Yeah.
Totally the same type of thing here. Like your inductions, the recipe for the inductions alters because if you've got like a wave bag, like it's which is really just a really giant Ziploc baggy bioreactor, your mechanical forces are going to be significantly altered. So, yeah, scaling is hard.
Cool.
Yeah.
And I guess just like another add-on was I'm just generally curious about like the B cell differentiation. So like if one wanted to like culture some B cells and like see if they could do some type of like evolution and try to get some like nifty antibodies or something like that. I don't know if you guys have ever seen that, or actually, yeah, like have you guys seen anything like that before, like any papers or just like kind of personal experience?
I know we're gonna talk about it a bit later, but yeah. So there is this research try to cure diabetes mellitus, and yeah, the B cells in the pancreas that produce insulin and control the glucose level are like yeah, they're tightly related to it. So yeah, if you're yeah.
I get that, yeah.
But as far as like the formation of unique products like antibodies using something like directed evolution, that just probably would not work,
Right.
from my understanding.
Exactly.
Okay. It's just tricky to get like I guess the somatic hypermutation to go and like actually get like directed evolution kind of.
Well, it's also that you're trying to produce a unique product, which typically, antibodies like that are formulated over many, many, many generations. So I don't know. Gabe, do you agree?
That's what I was
Yeah,
wondering. Like, how do you get the
that about nails that. Yes, correct.
Okay.
All right,
Well,
on we
awesome.
go.
So yeah, next. So embryonic stem cell can be harvested from the blastocyst. And oh wait, yeah, exactly. I was looking at the slide. So harvesting methods, the embryonic ones can be harvested from the blastocyst, which is like the state at 4 to 5 days after fertilization in vitro. This embryo, like the after fertilization, this embryo is now called a blastocyst. It has an inner cell mass that can be harvested, subcultivated, and or stored as frozen. These cells are not embryos yet, so the evidence emerging that these cells do not behave in the lab as they would in the embryo state has also kind of pushed the ethical, like lower the ethical concerns. So technically, we could at the moment where someone's pregnant kind of thing, or like at the moment they tested positive for pregnancy, you could have one cell being removed from the blastocyst and not leading to any complication later on, but that specific cell could be used to cure your own diseases later on. So yeah, adult stem cells on the other hand, they're found pretty much everywhere in the differentiated tissue that you have, like your bones, your ligament, your nerves, and everything. And there is no evidence of an adult stem cell developing other embryonic germ layers. So they don't go like from your muscle. They don't go to the pancreas and divides into a pancreatic cell. Like they stay where they are generally. Uh, yeah. I should stop saying. God damn it. Best practice. Okay. An adult stem cell will preserve from a limited time its amount of genetic material. Therefore, plasticity is also a key factor, finding the potential generating specialized cell. This could tell us the importance of embryonic stem cell having an extreme high potential. And the methods used to getting stem cell for researching purposes has been improved over the years. We can now harvest them without sacrificing embryos as conventionally were done and thought of. Scientists have refined quite a few applicable techniques that help get a deeper knowledge in this promising field. Blastomer extraction has a technique proposed by Robert Lanza, where a single blastomer cell is removed from an 8-stage division, and the blastocyst cultured from the embryonic stem cell is not harmed. Like you could go on through becoming a full human being without any problem, even though you had like one single cell removed after the 8 stage. Any questions?
Well, not a question, but a bit of a comment. So
Yeah,
this removal of a single cell from a blastocyst is actually often done in vitro clinics. When a
yeah,
family with a history for a genetic disease is having like an in vitro procedure, they would have like multiple embryos, and a single cell would be removed, and a PCR test will be performed to see if this particular embryo is clear of the disease that they are worried about.
yeah, yeah.
So yeah,
That
this
is a
is this
very
is
good
commonly
explanation.
done.
Yeah,
It's like a
yeah,
routine procedure. My mind was blown when I heard that.
yeah. Well, you described it quite well. I don't know how long we've been doing that. Again, I wrote this paper with some friends, student friends, like freaking 10 years ago. 20, yeah, no, actually 8 years ago. So yeah, the technique has been improved since, and I'm sure like they're doing it now. They're actually like keeping those precious cells for future use. Yeah. Any more thoughts, comments, or questions? And thank you, by the way. Thank you for describing it a bit better than I did. Okay. Description of induced pluripotent stem cells. acronym IPS or IPSC. Shinya Yamanaka won the Nobel Prize in Physiology for his discovery on how to transform ordinary adult skin cell into embryonic-like stem cells. It is therefore possible to induce pluripotency in adult stem cells that can genetically be reprogrammed to become embryonic-like and therefore pluripotent via inducing and expressing specific gene and protein factors. Adult stem cell can go back to the embryo state via regulatory genes, like four main gene factors, namely the Yamanaka cocktail. And this scientific breakthrough is pointing at a limitless, like near limitless source of immune compatible cells for transplantation or tissue engineering, and possibly even reaching consensus on approval for stem cell use in some countries. A patient's blood stem cell could become another tissue or making a new organ simply by traveling back in the epigenetic clock. Like the time travel described is basically like an epigenetic time travel, which is I can try to explain quickly what it means. You have altered methylation, which is like silencing specific part, and acetylation, which is a promoting specific part. So you open the histone the histone like the histone and chromosome, which are the ones that tightly pack them together. You can open them and close them in specific sites. So our reprogrammed human skin cells were virtually indistinguishable from human embryonic stem cells, said Plath, an assistant professor in biological chemistry with the Eli and yeah, well, basically like their center of regenerative medicine and stem cell research. This procedure is extremely risky, though, and genomic and epigenomic alteration that are abnormal will be likely to occur, and this is one of the reasons why iPSC cells have not been broadly and or officially used in therapy just yet. Next slide. Or questions, thoughts?
Okay, can I add another comment?
Yeah, go ahead,
I think
please.
it was last year that Professor Yamanaka had visit here in my country, and we had a lecture with him.
Cool.
And
Which country is it?
Bulgaria. He was here
Cool.
for
Very
like
cool.
2 days or something like that.
Okay.
And you will probably get to this in the lecture, but they have a really interesting approach on how to circumvent the quality control to be done for each individual person. Do you have this in the lecture?
Not really, no. So if you care to speak up about it, like by all means, it would be awesome.
Well, so the biggest problem with induced pluripotency cells is the quality control, as you mentioned, making sure that each cell has correct epigenetic programming that randomly occur, and
Mm-hmm.
so on. So what he said is
Does
was
not turn into cancer.
Right,
yes,
right.
basically
Just
this.
technically, yeah.
So what he said is that it takes his team like a full year to make sure that the cell line is stable enough, it doesn't cause any problem. So if you want to apply this in like medicine, experiment, practical, this is extremely expensive just because of the time that needs. So what they're looking into is they have basically checked what are the most common types of HLA proteins in the population.
Yeah,
And
exactly, yeah.
they have picked up, I think it was 30
Mm.
specific proteins, and they've located people which have only this protein, not a combination of two, like
Okay.
most have, but that have the same copy on both chromosomes.
Mm-hmm.
And actually, I'm not sure if it was 30 or 100. Anyway, they have this group of people which have only one HLA protein, which are considered to be like kind of like universal donor. They would be able to donate to their induced pluripotent stem cells grown from them to any person that has this HLA protein and potentially another one.
Right. Okay. So yeah,
And
that's basically, yeah. Okay. Yeah. Yeah.
and in their estimation, this would cover about was it 30% of the population in Japan? Those 100 individuals had the cell
Yep.
line. So that's a really interesting way to circumvent the entire full year
Yeah.
of quality.
Exactly, like the uncontrolled and like behavior that leads to abnormalities and cancer in the end. Yeah, yeah, definitely. Cool. That's basically like a better explanation on how he planned to make induced pluripotent stem cell bank factors kind of thing. I have still yet to read up on much of the latest studies. So thanks for sharing this information.
So people
Yeah.
who are interested in doing more of it. So I looked, and we're getting close to the end of this presentation. So this is a good time to talk about this. For people who are interested in more of a deep dive into Yamanaka factors, I think there's actually 32 or 36, something in the 30s. But OSKM are the four that everyone gets really excited about because they're basically at the top of the cascading pathway, which means all of the other factors get triggered. And if you look in the Yamanaka folder, there are constructs and papers about work that's been done recently. If you are interested in where Yamanaka's work has been picked up and taken further, I suggest looking at things that have been done by the Salk Institute, SALK, Salk. They were the ones who developed the toggleable switch that allowed Yamanaka factors to be turned on and off, because if you turn on Yamanaka factors, eventually you just get cancer. It just because
Right.
like that's what pluripotent stems, like totipotent stem cells, are in a fully grown organism. And so having the ability to toggle these things on and off was very important. And so if anyone is interested, the Salk Research Institute is the type of thing that you want to dig into. And like I said, the papers that we have in the Yamanaka folder and the constructs, some of which we have been testing here at SciHouse to see if we can have a TET-inducible toggleable switch, which allows you to turn the Yamanaka factors on and off. So there's more research there for people who are excited about this concept and want to dig a little deeper.
Right. I know, Gabriel, you mentioned sort of a dimmer that could be installed in order to, like, instead of 1 or 0, you have like a gradual controller to, like, increase it more smoothly and decrease it more smoothly. That may also prevent many complications. But
Yeah,
anyways,
exactly. That's
Yeah.
That's kind of the point of that.
Is that what the shit ton of Oxytet that you had was for?
Yeah.
Mm-hmm.
Actually,
So,
no. The oxytetracycline is for something completely unrelated, and I'm not going to go off topic. The doxycycline. It was the dox It was the TET-inducible switch, which also responds to doxycycline.
As the Andreas would say.
Yeah, that was that stuff with Yuri.
Cool. Okay, let's get through this paper, and then we'll have a little interesting talk later on, probably, hopefully. So differentiation pathways of inducing pluripotency, as thought of, back in 2012. Basically how adult stem cells operate includes a complex pattern of molecular interaction, intracellular signaling cascade, and gene activation, as mentioned, that can result in three type of differentiation pathways. So the nanotranscription factors display inhibitory properties in the formation of neuroectoderm tissue. You have SOX2 and 3 transcription factors that prevent human embryonic stem cells from differentiating into mesoderm multipotent stem cells, and thus maintaining pluripotency. The presence of BMP4 affects the OCT4 transcription factors, inducing HES cell differentiation into extraembryonic tissue. The absence of BMP4 has OCT4 inducing HES cell fate into ectoderm, and BMP2 to 6 heterodimer protein has been shown to mainly induce cell endodermal differentiation. So those are like those terms were what we found out in 2012. But many more proteins has been shown to influence the differentiation processes, and using techniques such as RNAi interference or retroviral gene deliverance to silence or activate genes is also possible to manipulate gene expression and thus direct cell fate into desired alternatives. Specific proteins can be introduced into the cell on the intracellular level, as well as the extracellular other factor that stimulates self-renewal or differentiation be introduced. So professional institution has proved to be like they provided researcher in that specific field to conditioned media. Like you need to have specific media in order to even like study those. And yeah, the description of somatic cell nuclear transfer is not covered here in the presentation, so you're welcome to look at the paper. But it's a procedure where substitution was proven unsuccessful, like not really. Like 1 out of 100 trials was successful, and yeah, so I'm not gonna cover that. But the potential future treatment, I think that's the next slide. Yeah. So Parkinson's disease, where the patients show similarity in the progressive degeneration and loss of dopamine and dopamine producing neurons, following the principle and hypothesis that stem cell could be successfully transplanted to produce dopamine. Diabetes, also, like we mentioned the B cells and the pancreas. Diabetes is a disease that is expected or was expected in 2012 to affect the lives of 380 million people by the year 2025, which has yet to be seen. But it's mainly a persistent and perpetual metabolism disorder, which is caused by the unbalanced number of B cells in the pancreas that produce and release insulin and control the level of glucose in the blood. Therapeutic aims of using stem cell as a treatment for diabetes is type 1 is to protect the remaining stem cell and replenish sufficient beta cells. Scientific and ethical progress in this field of stem cell regulation. You have fears that stem cell companies might now progressively open their business outside the United States, as you have like seen, and other countries such as in EU, like so they're basically moving away from too many regulation and a passive approach was taken back then by the US federal policy about stem cell research ethical issues. These issues were usually translated as limitation on taxpayer funding, scientific and ethical leadership, while protecting the so-called intellectual property interest and gatekeeping stem cell patents and licensing or licenses, so therefore prohibiting commercially sponsored research that could otherwise have been a realistic alternative to the federal funding gap. Japan getting ready for a possible IPS cell stock challenge. That's where Yamanaka of Kyoto comes in. He was a stem cell pioneer since the beginning, and he apparently still tries to break new grounds by introducing like this stem cell bank idea, like the fact of you could store like a protein repository that could theoretically, yeah, cure. He mentioned in 2020 like 80. Well, by 2020 he would have like a standard array of 75 induced pluripotent stem cell line that theoretically could be accepted by 80% of the population in the whole world. This was amazing already back then when we studied it and wrote about it in 2012. So I really should do some more research. I've been busy with other. This is amazing. Technically, yeah. If this succeeds, there is no much need for toxic treatment and so on. So back then, China was also disregarding stem cells rules unofficially, but officially they were like, yeah, we're doing our best to ban the clinical use of unapproved stem cell treatments. But again, it's I don't think it's that easy to regulate China, even for the Chinese government. So private clinics around the country were still treating patients uh, with money for like you know alternative treatment. Several private stem cell industry claim to have treated more than 10,000 patients uh, in 2012 with a variety of disorder using stem cell from aborted fetuses, umbilical cord, and adipose tissue, although none has published data from controlled clinical trials, or they might have just been removed, but temporary benefits has clearly been proven, which is still unclear if the injected cell would survive more than a few days in the patient's organ. So the ethical and social boundaries of stem cell research has some common religious beliefs that state that ensoulment occurs at conception, while other religious beliefs uh, mean that each of us began life as an embryo. So this is like a quick overview on the religion aspect of this, which also has a big impact on the ethical. It's many spiritual people would go hardcore to become like part of like ethics itself. So yeah, there's no secret. So
Yeah, and I think we have a bioethics folder
yeah,
somewhere floating
yeah,
around in the sorting.
right. So I'm not gonna go too deep on the Catholic and Islamic views, nor like the debate about abortion, because well, I can mention one sentence that what was already life that was going to be destroyed, and the choice of it being thrown away or used for research may may be a sentence that reaches some kind of consensus and ethical approval up there, because most countries are not against abortion, and therefore why being against embryonic stem cells. But anyways, yeah, the conclusion, I think we're there, right? Yeah. So induced pluripotent stem cell and human embryonic stem cell gave us an understanding that stem cell research might have bypassed one big ethical barrier by basically, instead of directly picking out one cell from the embryo, which probably is best, but we don't need that anymore. We can induce pluripotency in a fresh adult stem cell, and we only require a couple of transcriptional genes to get our cell and some epigenetic time travel. So, however, there is still a higher risk of forming cancer and lower risk of immune rejection. But that there is opportunities. Like I do believe in Yamanaka, but that's subjective. Everyone is entitled to their own opinions. Scientists may derive human embryonic stem cell to transplant more compatible cells, harvesting cells from the blastomere without embryo injuries, or simply prevent the zygote formation signaling of the SNT or with ANT. Unfortunately, the study of those has been subject to ambiguous skepticism in the US and Europe as well. So it has more or less been hindered in my place, but also Gabriel's place. Yeah, we'll see how it goes. For now, I'm hopeful. So a tremendous number of disease and injury could be cured via stem cell therapy, obviously. So we know, yeah, I hope that our understanding of stem cell has improved already here in our little club, but in the broader scale has improved enough to push it to support treatment for diseases that cannot be cured. That's it. Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
All right, let's open up the floor for some questions, clarification, and/or storytelling.
Alrighty, so I got here very late, so sorry if I'm going to be like repeating stuff, but have induced cells gotten to the same level of harvested embryonic and stuff?
I would assume it has a lower telomerase, a lower amount of telomere matter.
Telomeres.
Shorter telomeres for say, and a lower telomerase activity, but they're still very much potent in the terms of being able to differentiate into other types of cell and has conserved genetic material enough for being called an embryonic cell type. But
So like
I
the
need to do a
issue,
lot of homework. But yeah.
yeah, but like the issue then is they would. I know that's like the thing that controls when it goes into programmed death, right? So the cells wouldn't last as long, I guess, then, or what?
Right. So yeah, without telomerase and an adult stem cell cease to exist. But there is also like a high telomerase activity in cancer cells, but they have like a different mechanism of thinking that they're not part of your body. So it's ideally you would use those from the bone marrow, which has like still like is very much more fresh than, let's say, like a stem cell from your skin or from your muscle tissue. But then again, I need to do way more research on that for qualified definitions. Like I've seen much of the research on the mice leukemia, but that's it. I have yet to, as far as I understand it, yes, like I don't know who said it, but the induced pluripotent stem cell bank is still on, so I need to go into that and look it up in further detail. But yeah, anyone else has something to add to it?
Now I've got a question, but my understanding of this is also very, very limited because my interest in this is basically from sports medicine due to personal injuries and the obscene costs of stem cell therapies for sports medicine, specifically in tendon repair. Stitching two tendons together is not a good solution.
ACL work. Oh god.
Personally, it's medial collateral, not ACL, but same.
Yeah.
And I just forgot my question. Oh boy, here we go. Oops,
Like,
sorry
what do you
about
want to? What do you want to do with them?
Well, like, can it help with tendons and stuff like that?
Yeah, there's already companies doing sports medicine. So Joe Rogan has been incredibly vocal in his support for stem cell therapy, specifically for the one company that's basically offering.
Yeah, we're not going to utter the name.
Oh, okay. All right.
Sorry.
Is it kind
Sorry. Nope. We're not
Is
going
it
to talk about
pseudoscience?
Trump. We're not going to talk about pseudoscience and Trump supporters here.
of...
Oh boy.
yeah, they're nasty.
Yep.
Someone is doing cell therapy, I guess. Is
Well, so
I mean, I know I have a coworker that went down to Mexico because he tore his ACL, and he got that stitched up, and he also got stem cell therapy and some BPC 157. The whole works.
wow, and it helped quite a bit.
He had a really weird response to the anesthesia, but that's like completely unrelated to the fact that it worked. He's walking around just fine.
Sure.
yeah, it does
Is
work.
is he
Here in
playing sports
Canada,
ball
it's like
again?
25 grand for treatment.
Yeah.
He actually has a pet pig, and the pig has like the pig version of diabetes, and so he has to give it an insulin shot every day, and so he has to wrestle the pig so that he can stab it with the insulin shot. So
Whoa.
no, he's not playing sports ball, and I know that that's a really weird story, but sorry, he's
Okay.
back to wrestling pigs.
Yeah, whatever. That's what's important, right?
I guess you could say he's throwing around the old pig skin.
Sasha, you're fired.
You can't fire me. You never hired me.
Well, it sounds like sport to me.
Yeah. I was wondering if anyone else does like or has looked at like organ on and chip stuff. I know that's been getting more popular lately, and more people are finding iPSCs like useful in that context.
Chip stuff?
Can you define chip stuff?
Sorry if it cut out. Organ on a chip stuff. So like organoids or
Oh.
just like
Yeah. I was working on a bone marrow on a chip thing for a while.
And
Okay.
it was
Because
hell.
and so that's all about it's not just about having the stem cells, it's about harvesting the stem cells, because you basically like create an artificial bone, and then you have this like constant flow of stem cells coming off of them from this fake bone marrow stuff. And we had some 3D printed parts, but it didn't really go anywhere.
So you were trying
It's
to
a thing though.
you were trying to like make a bone and like have like a little bioreactor going and like making stem cells?
Like a tiny chamber, like a well that had a flow through it. Like it was a tiny bioreactor, but it was really about like making like artificial bone marrow built off of an amalgamation of stem cells that had built up, and then you have your like foamy core, and then they're like, hey, I'm going to be bone marrow, and then I'm going to start doing this, which would be really, really great for testing, because there's a lot of stuff that you want to test, and you're like, wow, if I stab this into a human being, it might hurt a whole goddamn lot.
Sure.
And so, you don't want to do that, maybe.
Can't do human experimentation like the old days.
You got almost buying a cow femur.
I don't know. All you gotta do is find the right person in the FetLife community and then get some.
Could it make blood?
Yeah, that's kind of the whole point, is because so this was for the testing of toxins and venoms, which meant that you would, like, you need to have blood and you need to see how things respond in an organic environment. And so it's like basically the intermediary step between in vitro and stabbing a mouse.
Hmm. So what? You would still keep it network bone as well. It wouldn't just be like a blood farm, or wouldn't just be a bone, but kind of replicating that system.
Exactly. So it had multiple stages, and it was kind of like a loop. We made a poster for it at the last Biohack the Planet, and I've still got some of the pieces floating around. It's not like it's not doable. It's just autoclavable, as everyone has learned from the Nocra project. Autoclavable
Resin.
resin is really fucking expensive. So that was kind of the wall that I hit with that.
Gotcha.
Could you like do like? I'm trying to think. Is like the plastic could like a metal be used, or would that mess up bioadhesion, or what would it be?
Really just want borosilicate glass. And I know someone who can do glass blowing, and I'm like, okay, I just want you to make lab equipment.
There's
Like,
an open source glass lathe. I don't know if I posted it here yet, but someone made an open source one. Once I make a like giant makerspace, they're just like making all the weird glassware. Oh, yeah.
So when I was in university, they had a glass blowing lab that was in the basement of the physics building, I think. And all the chemists and the physicists and the biologists would come, and they're like, hey, we're going to do something nobody has ever done before. And because we're doing something nobody's ever done before, we need a piece of hardware. And then they would get it done. So any of you who are in university, like proper university, like a big freaking university, see if there's a glass lab. There might be just a glass lab. There's a dude. He probably has a big beard. He's in the basement and he's blowing glass, making weird stuff for physicists. And then you would just come down and you would talk with the dude and you'd be like, yeah, I need to make this weird ass thing. And he's like, okay, well, this and this and this are all wrong. Your drawing sucks, but blah 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 blah. And then he would just
Make
he
it
would
Babylon.
make it, and then he would just be like, yes, and it would be perfect and beautiful. And working with borosilicate is really, really hard. And so, of course, these people are extremely talented. So their glass work is perfect. And that's really what we need.
I think there might have been a glass blowing YouTube channel or two. I don't know if they were like what's the word? There's a word for that profession, like scientific glass blowing or whatever. I don't think they're that level, but definitely.
Well, Baraximus, the person Baraximus, he is a really great glass blower and a friend of mine, and he's just busy trying to become a surgeon in New York City during plague.
Oof.
So so he's busy, but I still think that maybe it's more about finding the hardware. You know, you just you need the kilns, you need the big freaking stuff.
But also for the Nocra project, there could be a workaround, since the base of it is kind of not even technically the main material that you're working with. Because, I mean, they kind of used plastic before that I don't even think was technically autoclavable, and then did all the chemical mods to it. So in theory, you could do like a coating after maybe autoclaving it and then just pour it into whatever mold you want.
This is more about having like a reusable mold.
Yeah,
We've gone off topic. Let's get back. Oh my God! Why do we always end up on Nocra? Okay.
All roads lead to Nocra.
I was
Yeah.
just
Okay.
about to
So
ask you why the plastic needs to be autoclavable, but yeah, later.
because I want to use it more than once.
Also, contamination.
Yeah.
Okay, I guess I'm one of the rare lucky persons. We have a plasma sterilizer in the hospital.
Yeah.
And I've done some
Because you're in a
testing with,
hospital.
yeah, I've done some testing and PLA prints survive it without any damage. But I guess that's not available everywhere.
I could, in theory, purchase one of these objects, but I don't have enough money. They're very
Didn't-
expensive. They're like
I'm
stupid
pretty sure
expensive.
it's like entire room.
Uh-huh. Yeah.
Anybody
And
Bitcoin millionaire here? That help a little bit.
any Bitcoin millionaires, we're looking for a plasma sterilizer to make knives made out of bacterial created abalone shell.
Is this the cold plasma thing, or is this like electron beam irradiation sterilization?
I have no idea. I haven't asked them. I just know it's plasma something.
Not
low
practical for
temperature.
biohacker applications.
Because I know there's like the
Yeah.
little torch thing the Thought Emporium or whatever made, and then there are like the things you use where it's literally like vacuum shooting electron beams at stuff. Like craziness.
It's more of a novelty. It's not super practical for sterilizing large shit.
If it's hospital, it's ethylene oxide.
Ah, okay.
Yeah. Okay, so back to the topic at hand. Yamanaka factors. Anyone got any question?
No.
Yeah, I think I'm relatively good. I asked my questions, I think. Oh, and I posted a link to a TED Talk I saw forever ago on application for Alzheimer's, I think.
Yeah.
Of course, some of those can get kind of sketchy with who they let on, but I think it was one of the kind of legitimate science TED Talk things.
Yeah, guys, if you have anything you're willing to share, or like, there is also like a whatchamacallit, a text channel, like the Yamanaka text channel. Dump all you want in there that is related to Yamanaka. I will be super grateful. I think if like, all Sci House that is interested in it will be super grateful. So if you want to share something, just shove it in there, and we'll applaud you internally.
Yeah, and it's only been finally maybe it's only been 6 months since I was last working on a Yamanaka project. So if anybody has any questions, I am more than happy to answer them. And again, please dip into the library. There should be papers and constructs of things that were done, how these things work, integration, all of that type of stuff. So really just, if you want to go down that rabbit hole, I'm here for you.
Oh, also for anybody that's interested in like the weird drug therapies relating to the stem cell stuff as well, there's this really cool research going on at the Mayo Clinic in Jacksonville, surprisingly, that works with extracting extracellular vesicles from blood plasma and then loading that with whatever therapies that you're using. And they also use combinations of stem cell therapies as well. So that's interesting if anybody wants to look into that.
Awesome.
I've got a rabbit hole to go down.
Yeah.
Most
and this may be a naive question, so forgive me if it is. So a bone cell is a bone cell is a bone cell, right? Right?
If I understand you, yeah, I think so.
Okay.
Sure, let's put that as a basis.
So how does a bone cell know that it's a tibia bone cell and not, say, a skull bone cell or a tooth bone cell?
Because of the stuff that's around it that causes the induction.
Yeah, structural and chemical induction.
So it's not in the DNA. It's
so every cell has all the data. That's the thing.
Yeah.
Every cell has all the data. It only expresses certain things based off of physical interaction, chemical interaction, and mechanical interaction. So how does the bone cell know to be a tibia and not like randomly make a skull in your hip? It's because it's in the tibia structure. And so it, like, this is how the ghost heart project works. This is how you do ghost hearts. You take a pig heart, you decellularize it, you've got this floppy matricey of heart shape, and then you seed it with stem cells, and the stem cells are like, wow, I'm in the heart area, I will do heart things now. It's actually pretty freaking fantastic.
Yep, this is like a Bio 3 thing too, because I'm just getting into that in my coursework, and you'll find out that there are basically two ways that any sort of organism differentiates its cells, and one of them is just by gradients. So like simple organisms like flies do it just by making chemical gradients, and they're like, this thing has more than that thing. This is going to be the end of the fly, or the induction. Like good example of that is you can't grow a lot of organisms in 0G because they don't know which way is up or down, and the cells get confused and turn to cancer. So, yup.
Yeah, I've had my coursework for a master's degree on something similar. Well, it was kind of boring, sequencing, but it was from patients with aniridia. And their issue is that on a, let's say, middle level, most of those gradients are created by the products of genes like maybe you've heard the homo box, HOX one,
Yep.
2, 3, something, something, and paired box,
Love me some
PAX.
Hawks genes.
Yeah, so with aniridia, the gene that breaks is PAX6. And from what I remember, PAX6 and PAX2 form gradients that go in different directions. One is in the frontal part of the eye and the other in the rear part of the eye. And they go against it. And that's how the cells figure out what they need to become, whether it's high PAX6, low PAX2, and the opposite.
Yeah, there are actually interesting control mechanisms in vertebrates for rib formation. So interestingly enough, in snakes, the genes like run parallel to each other, and it basically says form all of the ribs. Versus in humans, obviously, only part of the section has overlapping that causes the formation of ribs from vertebrates. But that is also pretty interesting.
Oh, I had this recollection from first year in my bachelor's degree that actually such repeating patterns are created by sort of interference structure between several factors. I don't remember that clearly, but it's something like that.
Yeah,
Basically,
it's
two
two
gradient
interference
sort of
genes.
interference structures, high and low points.
Yep. Any more questions? All right. Thanks, Sozik.
Yeah,
Awesome.
thanks,
Thank you
Aniki.
for being
Thanks,
here.
man.
Thank you.
Yep, thanks. It was good, even though I was very late.
Oh, it was dope. My pleasure.
Thank you very much. I haven't had a lecture in 2 years or something. That was refreshing.
Yeah, that's why we're here.
Well,
Like
we
we're
do.
combining lecture and hanging out. I enjoy it a lot.
Yep. We'll be doing Journal Club every Wednesday.
Hell yeah. Yeah, I hope I was kind of more expressed, more verbose and clear enough. Like it, I hope I'm improving. Because yeah, the course that we're having, I really need to stop saying I need to like have I have a couple of bullet points that I need to focus on in order to be a better speaker, and I think
Yeah,
I'm improving.
it's also not your first language, so it's okay. It's the thing. So and if you're from the west coast of America, the word like, and
Yeah.
if you're from France, it's the
Yeah.
is